Alors maintenant, on passe en visio. C'est la première visio, donc on va voir tout de suite si ça marche ou pas. Donc c'est Damien Huzard qui est à Montpellier, à l'Institut de génomique fonctionnelle, et qui va nous parler du toucher affectif chez la souris.
Bonjour, vous m'entendez?
C'est bon.
Oui, c'est tout bon. OK. Bon. So thank you very much for the invitation. And today I will talk about a little project I'm working on at IGF in Montpellier, which is about manipulating affective touch to study the social deficits in mice model of ASD. So what we are trying to do is to use a specific kind of cells that are in the skin called the CLTMRs, which are responsible for the affective and gentle touch in mice. So the CLTMR stand from the C fibers, which are unmyelinated with a small diameter and a slow conductivity. And the LTMR means that they are activated by small mechanic pressures on the skin. So those cells have some equivalents in the humans, which are called the C-tactiles afferents, and they have been named the gentle touch fibers. So to study them, it is possible to use the microneurographic recording, which means putting some electrodes directly into the nerves of the arms and recording and manipulating with a brush and on the arm of the participant, and people can record the firing of those specific cells. So the important part here is that doing some psychophysics measurement, it has been shown that there is a specific velocity, which is around 3 centimeters per second, when the touch of the brush is felt the most pleasant. And it is actually when the C-tactile fibers have the highest frequency firing, and it is linearly related with the pleasantness. So this really means that there is a cell in the skin that is encoding for the gentle touch. Now it is important also to make the link, for example, from projects which are working with preterm babies and applying some skin-to-skin contact. And the project I'm involved in is trying to figure out if it is really the C-tactile fibers that are encoding the social touch occurring during a skin-to-skin contact, which will increase the pleasantness, some parasympathetic increase with probably the activation of the vagal nerve, and relieving the pain, and then attributing some reward and creating the bond between parents and the babies. So our goal in the lab is to try to create a model in order to manipulate those CLTMRs in mice. So we use the combination of different channels, the calcium channel and the sodium channel, which when we combine both with some Krylux genetic recombination, we should be specifically targeting and removing the calcium channel in the CLTMR, meaning that we are reducing the activity of the gentle touch fibers in the mouse. So we did a collaboration with Pascal Fossa in Bordeaux, and they did in vivo electrophysiology. And they applied a small pressure with a Van Frey filament, a little filament on the skin, and they recorded the responses of the CLTMRs in the skin. And we can see below in gray that the knockout mouse do not have the response in the CLTMR. And they also recorded the activity of those fibers and found that the fibers which have the deficit in responses are the slow fibers, which are the C fibers. So we should be targeting the proper firing neuronal population. We then studied these mice in some paradigm to study sociability. So the first step that we did is a 3-chamber social interaction test in which a mouse is freely allowed to explore a maze. And on the left chamber, there is a conspecific, so another mouse, or on the other side, there is an unanimated object. At the end of the test, we are computing a preference index, checking whether the mouse is spending more time sniffing a conspecific or an animating object. And what we observe is that in white, the knockout mouse spend less time sniffing the conspecific. So this illustrates the decrease in social preference in our mouse. So all these characteristics has been summed up in a paper that we sent under revision. But now we are also trying to target a bit earlier and see if we could do some early intervention in those mice. So what we did is to replicate a protocol that has been done in 2013, in which they showed that in human, for example, when a mother is holding or carrying the baby, this could have an effect on the heart rate and maybe the vagal component of the heart rate. So in red, you can see here this increase illustrates a decrease of heart rate. It is the interbeat interval. And by doing the same on mice at P10, so really early after birth, it is possible to mimic the holding and carrying, showing that holding has the capacity to reduce the heart rate. And this is what we did in our animals. And we tried to do some resting or carrying condition. And interestingly, the only difference that we observed was that in the mice in which we removed the CLT-MR, they have a higher resting heart rate. So we were actually surprised to see that the decrease in carrying was not significant, but we might have a deficit in the basal capacity of the individual to activate the vagal nerve to control the cardiac rhythm. So those are preliminary results, and now we are trying to add some experiments to see whether we can manipulate it or not, and see if this has an effect also on the social behavior that we observe at adulthood. So thank you very much for your attention, and if you have any question or even advice for the future experiments, I'm more than happy to take them into account. Thank you.
Merci. Est-ce qu'il y a des questions? Bon, on n'a pas de questions. On va arrêter là la matinée. Merci beaucoup.
Merci.